is called monitoring grouper songs, the use of hydrophones for monitoring fish reproductive aggregations. The objective today is to present the use of hydrophones to monitor fish spawning aggregations and share results of research conducted in the Sian-Can Biosphere Reserve and the Arrecifes Ixcalac National Park in Mexico. We will share with you that the information that we have interpretation available, which you can see on screen. The instructions are on screen on how to access it. You have access to the English language channel controls on Zoom. If you're connected through Zoom, you can listen to the audio interpretation in English or in Spanish if you prefer. You can also turn off the original audio if you wish to. Some basic rules of this seminar are a space for learning and respect is encouraged. Microphones and cameras will be disabled during the seminar. Please enter your full name, country, and organization in the chat box so we can have this information registered. You can also post your questions in the Zoom chat box or the question space. If you participate via Facebook Live, you can send your questions through the Facebook comment section. Your questions will be taken up and answered by the presenters at the end of the presentation. The opinions expressed in the webinars are the sole responsibility of the speakers and do not necessarily represent the opinion or position of MARFund. Once again, welcome, and we thank you for joining us on our 14th session of MARFund seminars called Monitoring the Song of the Grouper, the use of the hydrophone to monitor spawning aggregations. Our speakers one of them is Jacobo Kamal Madrigal, who has 12 years of experience in natural protected areas and working with sustainable fisheries in Mexico in collaboration with managers of natural protected areas and fisheries resources, fishing cooperatives, government agencies, universities, etc. He has 9 years of experience working with refuge zones and fish spawning aggregations in the Mexican Caribbean with a strong focus on community participation, including training people to execute biophysical monitoring, database management, and analysis, project management. Our second speaker today is Aniela Ancona, who has been a monitor diver for 6 years, member of the Community Center for Scientific Research and Underwater Monitoring CESIMS of the SPP Pescadores de Vigía Chico de Punta Al in Quintana Roo. She has experience in reef monitoring of coral reefs. She has been participating in monitoring fishery refuge zones and has trained colleagues for other locations. She has participated in the monitoring of spawning aggregation sites with visual census, environmental DNA collection, and passive acoustic monitoring in 3 spawning sites in Quintana Roo. She's part of 2 thematic groups of a network of fisher women and fishermen in which people from the fishing sector from different states of the Republic participate. And she loves to think that the sea is a friend, that we don't have to fight against it, but we simply have to share the same rhythm as it and that will help us move more freely within it. This is additional information to contact us through the different social media and our webpage. And now I will give the floor to our speakers. And I am Michelle Fernandez. And if you have any questions, I will be keeping an eye on the chat box on Zoom and Facebook for your questions. Thank you. And here is our first speaker. Hello, good afternoon. I will talk to you first of all with one of the activities we carry out in Punta Allen called the passive acoustic monitoring. We are a small community in the state of Quintana Roo called Punta Allen. This community is located within the Siancan Biosphere Reserve. This reserve has 2 fishery refuge zones since 2016. We have 2 grouper spawning aggregation sites that are protected. These are protecting groupers. The initiative was proposed and promoted by a society or a cooperative called Sociedad Cooperativa de Producción Pesquera de Pescadores Vigía Chico, the fisheries of Vigía Chico. Today, we now have a community center for underwater research and monitoring, which we call CSIMS. There are 3 women and 8 men that are part of the group. There are fishermen, there are housewives that are part of it, and tourism service providers. And the group arose from the need for information to benefit local fisheries. And now we are backed by the Cooperative Society of Fisherers of Vigía Chico. This is a timeline you can see below, where in 2015, there was an open water dive where we also carried out first aid, barometry, and exploration, ARP exploration. Back then, those were people involved that are not with us now, but in 2016, 2 people that were certified became part of the team. Then in 2017, we had a specialty now, and there were more people taking the open water. And in 2018, there was the hobos handling passive acoustic monitoring. And this was where we formed a group of CECIMs. 2020, there was a course of connectivity and of water and how to take tissue samples, what we call the environmental DNA, with 9 members. And in 2020, 2021, where, as you know, most of us were left without activities in the field because of COVID. This happened. But in 2022 and 23, we returned to passive acoustic monitoring and data analysis. And here you can see that the first 2 lines from top to down are the current members of the CECIMs who are active members of this team or this group. The people below, in the line below, were the first ones to start taking the course, but for some reason or other, they're not participating anymore, but the majority are still part of it and are benefiting all the efforts of the community. What we do is basically we monitor a reproductive aggregation or a spawning aggregation of fish where there is a concentration of fish or specific groups of fish where they congregate very predictably and repetitively with the purpose of spawning, happening in moments and specific cycles. That's why we have localized this, when we know what time and place they're going to be with us or getting together. This is what we do, a passive acoustic monitoring. This device, which is called a hydrophone, what it does is basically records marine environmental sounds, and incredibly, it does this below the the sea. This hydrophone records 20 seconds, sorry, of audio, and then takes a break of 300 seconds. And the activity is done from 70 to 90 days, and done between December and June, and with a depth of 20 to 30 meters is how deep is located. And we locate them in 1 to 2 dives per site. It is an acoustic recorder which records all the underwater sounds. It does not record specifically one single sound or the sound of groupers, which we are monitoring. It basically records everything that's found, anything that makes a sound underwater is registered by this recorder. And that is where we focus and take more attention into what we are focused in, which are the groupers.
So after that, we do an analysis of all the information, and we extract the information from the hydrophone, and we take out the hydrophone, we discharge it, and we save it on the computer, and we keep a backup too, and then we do the research again. Perfect. Thank you very much, Daniela. I will continue the presentation. Thank you, Jacobo. While Jacobo connects, I will remind you that we have simultaneous interpretation for English speakers, and we continue with Jacobo. Thank you. As Daniela mentioned, I would like to stress that this is an activity that we do it in collaboration with our organization, which is Community in Diversity. It collaborates with people in the community to collect all this information. They've been trained for a long time. They collaborate actively in this activity. After what Daniela mentions about collecting information, all this information is downloaded and is passed through a program, which is an algorithm that what it does is that from all these sounds of the marine environment, it isolates the particular sound of 4 species, which are the Epeniphasis stratus, the Iteperoperca winasi, and venenosa. And so we have isolated the sounds of these 4 species that actually spawn in this site. We obtain this information. On the screen, we can see that the program appears, and this has been facilitated by Dr. Laurel, who collaborates and has shown us and taught us how to use the program, that we are very thankful for his collaboration, because there's a lot of information that we have been able to collect. And so all the files are of 20 seconds are there, and the program interprets the information as such. In this below, we can see the total of hours that we have collected the sounds in different sites. I tell you that in Quintana Roo, there are 3 sites that are verified visually that are spawning sites. Some are from one species, others are from diverse species of grouper. And this is what basically the program does. It processes it, and it tells us in each file how many sounds of grouper we are detecting from these particular species. And once all this information is analyzed, it is collected in a file, and we ask them to give us this Excel file where we see the particular file where we see if any sound has been detected, as you can see in this example. And here it says that there was one particular sound that was detected of a certain species. In this case, it says it's from the Estriatus species. And this is what is done. And so basically what we're doing currently is we're using Excel to be able to have a basic interpretation of this information. As such, we know there are other ways of analyzing in a more detailed manner, but at the moment, we are only doing it with a particular Excel. And this graph is from a particular community where Agnella lives, and it is, we see here a graph from 2018 to 2023. So I must stress that during the time that we have observed this and the sounds have been registered were during the month of January, February, March of each year. The experience that we have had is during those months, and this data, first-hand data, help us to understand if the fish are actually arriving there during those dates for spawning. The gray areas show us the full moon of each one of those months, and the red colored color shows us the days where there is a greater amount of sounds registered, and this is linked to the events of spawning as such. The fish in general, when they reach the sites, spawning sites, we have the reference of the full moon and the days after the full moon is when the fish start to arrive in greater quantities. So this is a general vision. We have data from approximately 2008, 2019, 2020, because of the pandemic. In 2021, we were not able to have data, and so it was a little bit complicated, also 2022, but we were able to obtain some information. This graph in particular is only for the Epiphyllus species, which is specific to that site. This other graph that we see shows us that what we did was entering the data from the different years, focusing at this time mainly on the month of January to be able to obtain an example of what this information can tell us. The gray circle in the graph represents the day of the full moon, and we can see the numbers at the bottom of the screen, which are the days after the full moon. Let's say if the day was the full moon day is represented with a 0, and this graph helps us to visualize it, whether even though during January the fish are arriving to that particular site for spawning, it also shows us that it doesn't, the events do not always occur at the same time with the greatest registry of sounds. For example, in 2019, on the 7th day after the full moon is when we registered a greater quantity of sounds. And in 2018, it was around the 11th day, 10th day after the full moon when the greatest amount of sounds were registered. And this, the day where the greatest number of sounds were registered, we might have been the day we think when there was a greater spawning activity. We have found this, and this is how we find this. And this makes us understand, leads us to understand that if the spawning every year takes place in January, every year the behavior is not always the same. We do not know exactly why this is so. The temperature of the water is very important with the spawning, also the currents, and these variables could be determining the reason why during some years the days after the full moon, there is greater registry, and others there is less. We have data here from 2018 to basically to 2023, which fortunately, and in spite of the circumstances, we have been able to obtain information during these years that has been able to help us to interpret what is happening in the grouper uh, with groupers. And this graph, well, we've tried to play a little bit with the data. We see it's always the same month of January in Nietzsche Habin, the same data has been obtained from 2018 to 2023. But what we have done now is to try to understand if if it's during the same days where there is a lot of acoustic activity. So we aligned all the peak days where there is a greater registry of sounds, and we made the graphs with these, and we took the peak day, which is in the middle of the graph, as a reference, and we see that there is a variation in the quantity of days during which there is a lot more activity. As you can see, this is, let's say, the day zero, there's a quantity of registration, and then as the days after the full moon, there is also a larger amount, and this doesn't happen always at the same time after the full moon, but we did try to see if it is the same amount of days where there is a greater amount of sound. And what we noticed is that there is a variation among these days. For example, in 2018, we can see that there are about 4 days where there is a greater activity, but in 2019, it seems that there are a few less days, and this also happens in 2022. And this also helps us to understand that something is happening, that there are indicators in the site that in spite of the fact that they return every year in the same month and it's linked to the full moon, it does not happen exactly at the same time every year. And that they are also depend on the variables in the sites. This graph, what we did, and the research tells us that during the day, there are two moments in which there are moments of spawning as such, where the fish actually let the gametes out, and it has to do with the sunrise and sunset. And the acoustic information helps us to reinforce this information. At the bottom, it's the times or the hours of the day. And we also have the number of detections on the other axis. And as we can see, between 4 a.m. and 8 p.m., there is a small peak. There is an increase in register of that. In this particular site, the peak or the increase of sound during the day is more intense. So that means that we understand that during the afternoon, there's a lot more activity, and this is linked totally to spawning. This information helps us directly to be able to plan in a more strategic way the dives, to be able to have the visual information. And we were doing 2 or 3 dives during the day to be able to see when the fish was spawning. And with this graphic information, we know when we can actually time the dives during the specific hours in the morning or afternoon when there is a greater quantity of uh, sounds registered. And that is the first usage. And it also helps us to understand temporary what is the behavior of the spawning aggregations. And something interesting we can also notice is that in spite that from the point of view that the amount of days varies, at least regarding the times of the day or the hours of day, it is the same time lapse in spite of the fact that every year the quantity or the amount of sounds vary year by year. We can't assure at this precise moment that it means that every year each year there are less or there are more, but we can tell what is the maximum registration of sound every year. This other graph, what we were able to do from 2022, we started to replicate the acoustic passive monitoring in two new sites. Nietzsche Habin is one of the sites that we have data from 2018, but unfortunately, we had not had the opportunity to work in the other two sites. But after 2022, this became possible, and the community monitoring group called CESIMS have actually helped us to transmit the knowledge in other communities and to help participation. And this is a graph where we can have a simultaneous monitoring at this in these sites, and we can obtain data to be able to learn what is the behavior. Unfortunately, because of climate conditions, we were not able to obtain the same amount of data during the year in the three sites at the same time, but at least this gives us an idea of how the behavior was. In Nietzsche Habin, which is the green line, we can see that there is a large amount of sounds registered. And this could be because in this site there are more groupers than in the other two sites. But it is also interesting to see that how in 2023, for example, this varies, the registration of sounds varies in Nichavin regarding 2023. In 2023, the number of detection was actually a lot less in comparison. Unfortunately, for 2023, we were not able to obtain data. This is also proof that nothing is infallible because the microphone had a problem that was actually caused us to lose the data from February and March. So we were able to obtain from El Blanquizal, which is another spawning site of a grouper, and the behavior was the same. The behavior very similar, at least in the three sites, even though not with the same amount of registration, but very similar. And this is what we have up to now with acoustic data. This is just a very basic analysis that we do with Excel. And we know there are other ways of doing this, but at the moment, this is how we are doing this. And we hope that soon we'll be able to have more detailed analysis. And one of the objectives is to determine through acoustic passive monitoring in the very near future, what we will see next.
In 2023, we had the opportunity to conduct passive monitoring, acoustic monitoring, visual census, because what we've done before we have not been able to do this before, visual census together with acoustic monitoring. One advantage, though, for acoustic monitoring is that it has an autonomy that allows for 60, 70 days of constant monitoring where we can have information of 2 moons compared to visual census where we have to have a daily monitoring, fish counting, which is wonderful, but climate conditions sometimes are very difficult. the idea is that soon we can have a relationship of the amount of fish to be able to relate it with the amount of acoustic registry recorded and be able to have a relationship that can be able to give us a better number for the acoustic passive monitoring. But uh, because of the weather, this is sometimes an issue, and we know that nature is very wise, but these dates have the advantage of protection and make it even more difficult to reach these sites. So this is part of the information we have been able to receive, and we hope that in a medium to short term, we can be able to have the construction of models to be able to have a better acoustic passive monitoring. And this is what we have presented for you today, and thank you very much. Thank you, Jacobo and Aniela, for joining us and sharing with us today your presentation in this webinar. If there are any questions, you can ask them in the different uh, accesses we have through Zoom, through the chat, through Facebook, the comment section. For Jacobo and Aniela, we have two questions so far that have been written in one from Facebook Live. Micaela asks, is there any information regarding the temperature of water in the depth during monitoring? And yes, we do this. We do have this information. Thanks for the question. Since 2018, if I'm not mistaken, one of the sites is collecting temperature data and sea level data with a sensor. Since then, we have been collecting this information in the site of we do collect this information in Blanquisal. We began collecting since last year information of temperature and sea level in these aggregation sites. So we have some data regarding the periods of passive acoustic monitoring. We have depth of about 20 to 30 meters of the sensors, which is the depth where groupers are in activity. Thank you very much. A question is from Carla Mendoza, is what is the model of the hydrophone you're using? What type of hydrophone are you using? It's a Snap model. The Loggerhead brand has 3 batteries and gives us the autonomy of around 60 days because that's as far as we've left it because we have learned in the past that there are issues and if the battery runs out, which is about 80 days, we can lose data. So we try to avoid this by collecting the information sooner and rather than later and avoid to lose any data. Okay. So far, those are the only questions, but I have a question. When you presented the last slide, you mentioned the relationship between the number of groupers and the number of sounds. What is the number of groupers you have been able to relate to the number of with the relationship to the sound? It's very few moments when we are able to carry out a visual counting or visual census of fish when the hydrophone is installed. So the last few years, around 2019 to present, this has been very difficult to be able to do visual census because of the climate conditions which have changed enormously and have not allowed us to be on the site when the event is happening. Because precisely when this is happening is when the climate is worse, the weather is conditions are worse. With the hydrophone, however, is installed before the full moon, and the ocean is in better is more calm. But we have had situations when we have to change of battery. The weather also has been an issue for this situation as well. Thank you, Jacobo. The other question is from Raimundo Santisteban. What are the problems that you most commonly face in the field, and what are future plans? Aniela, if you want to help us answer the first part of the questions, what are the major issues presented in the field? For the activities regarding monitoring, the main issue or more complicated situation that we haven't been able to address is the weather, is climate. We initially began doing visual census, and the weather has not allowed us to have access to this anymore. But fortunately, we have the hydrophones, and which allow us to collect in a more accurate manner the information that we are monitoring. For Punta Allen, this has been the main issue, but we can't do anything else but adapt. Yes, precisely what Aniela just mentioned. We have uh, reached the community with a 5-day visit to do a visual census, and none of these days have allowed us for this monitoring, which is an enormous resource invested and very little results in the visual aspect. We have not been able to do more than wait for an open opportunity to be able to have this information. But the future plans are to be able to continue as much as possible to continue collecting the acoustic information, and obviously as much as possible to enrich it with or make it more robust with visual census. And we are in the process also of leadership transference to the community. The community of women and men divers are going to be responsible of going into the field now, installing the hydrophones and collecting the information. Now, it's not going to be necessary only for us as civil society employees to be able to do this activity. The community is in the process actually of training and have formed a community investigation group and are backed by the fishery cooperative. And their plan is to be able to uh, lead all this participative monitoring. And we are helping provide the necessary resources for them to carry out their activities. Thank you, Jacobo. And Aniela, we have another question. What is the characteristic of the grouper sound? And have you been able to perceive any other characteristic sounds or interesting sounds in these areas? As we mentioned, this program or the algorithm provided was developed by investigators who focused on identifying the sound of groupers, the different sounds, because there's more than just one sound. And once they identified this particular sound, what they did is create an algorithm to be able to detect this particular type of sound of these 4 species that we mentioned. And as we mentioned, the hydrophone doesn't record only the sound of groupers, but the whole marine environment. But the program detects the sound of these four species in particular. The other question by Sebastián Muñoz is, how did you have access to the reference sound for each of the species, kept the field recordings? And there is a second question, but let's answer the first one first. So those creating the algorithm also conducted the field work of verifying that the sound was in fact the sound of a particular type of grouper. It's a experimentation process with a hydrophone, and they verify visually that what is being recorded is a grouper. So this information was done with also with species under in captivity and in a laboratory, but the creation of the algorithm was done also with field investigation, of course. And this these are in fact the sounds of groupers. An interesting fact is that the sound which the algorithm was trained with was of a very particular region. And the first time when we received the program, the software, and we started to run this software with the data in it, we noticed that it was registering a type of species that with visual senses had not been registered before, and there was a very high number. So we started asking ourselves, where are these species? Where are they? And we shared this information, the sound, with the investigator of the sounds of this community. And he told us, surprisingly, that the algorithm had been having a mistake because the sound of the groupers of Punta Allen and the sound of the groupers where the reference groupers had a variation, and now they updated the software, and now the update understands the particular groupers of Punta Allen have, and now even though the groupers are of the different species or of the same species, so even those vary in sounds.
Gracias. La segunda parte
The
de la
second part of the question says that if do you have any data of referring these sounds? You have data on sounds overlapping with other fish species, percentage of error of algorithm when detecting specific sounds of the grouper species. The groupers have a very particular frequency, and each species has a particular frequency. And the program that we use is called Farad. Also gives us the option that we can obtain the spectrograms, which are basically the image of the sound. And we can see in those figures, they tell us what is the frequency and how the sounds, the waves of sound move in different circumstances. And it is complicated that there can be a confusion of sounds among the species because they're very particular. The program is trained to be able to detect that frequency, those frequencies in particular, because the frequencies also vary among the species themselves. Good. So I think those were the last questions that we have received, and we thank you very much for being accompanying us during this webinar. We thank Jacobo and Agnella very much for sharing their experience and their research. It has been very interesting from Quintana Roo, and we hope that this has been very much learning for our listeners and viewers. And so we would like you to please help us with a little survey for feedback regarding this webinar. The link of the survey is on the chat. You may access it now, and we will be very grateful, and we are grateful for your participation. Any questions you may have after the webinar, you are welcome to also ask any question in comments, and we'll take notes, and we will send the questions to our speakers. Thank you all very much. Thank you for the invitation.